Welcome to EPG Pathshala and to the first of 4 modules on sequential clinical trials. Thus far, we have been considering the standard clinical trial, where we have 2 or more groups who have been randomly allocated to some experimental treatment and to some comparison group, which can often be a placebo. We have seen that there could be different kinds of outcomes depending upon whether the outcome is continuous, binary, count, survival, and so on. Then we can apply standard tests of hypothesis with pre-specified levels of significance at the end of the trial to determine treatment efficacy. In this and in the next 3 modules, we shall consider a slight deviation from this protocol. That is, we shall assume that patients are coming in sequentially, one by one, and because patient enrollment can occur gradually and over time, as the trial progresses, we also have accumulating levels of evidence about the difference between the experimental and the standard treatment. From ethical considerations, say for example, a very strong difference emerges at the very onset of the trial. In that case, it would not be ethical to continue the trial if it is possible to allocate more patients to the better treatment. However, note that there are scientific issues involved because the size of the trial has been determined from some pre-specified power conditions, and because the trial is yet to be completed, we have not yet arrived at this power. Thus, there are issues of type 2 and type 1 errors, and it is necessary to develop a uniform set of methods to address this. This is what we shall explore in this and in the next 3 modules. Learning objectives for this module. We shall first introduce the concept of parametrization and define a parameter theta such that theta equal to 0 represents that there is no difference between the experimental and the control treatment. Next, we shall define an associated sampling statistic based on which the sequential plan will be implemented. We shall define a stopping rule which determines whether or not we have sufficient evidence to conclude the trial and see that there are different classes of stopping rules. Finally, we shall briefly look at some properties of sequential designs, and we shall revisit them in the next 3 modules. Sequential clinical trials, the first of 4 modules. The material in this module is largely based on the paper by Whitehead, which appeared in Statistics in Medicine in 1999 and is titled A Unified Theory for Sequential Clinical Trials. As is suggested by the title of the paper, in this paper, the author tries to provide a unified platform and define some fundamental concepts such as the parameter of interest, the statistic, the stopping rule, and so on to unify diverse theory which have been implemented in practice for the sequential clinical trial. Our framework here is that we have 2 treatments: the experimental treatment, which we shall denote by E, and a control treatment, which we shall denote by C. So the treatments E and C are to be compared using a phase 3 randomized clinical trial. Our objective here is to determine whether E is more efficacious than C. This conclusion can be drawn based upon some single patient response. In this paper, we shall focus on frequentist analysis and try to arrive at a p-value and point and interval estimates of the advantage which E has over C. The procedure here will be derived so as to satisfy some pre-specified power requirement, and the design of the trial will be conducted as a series of interim analysis. Interim analysis, of course, means that the data is to be analyzed at several time points in between the start and the conclusion of the trial, and we shall spend more time on the implications and necessity of interim analysis in our next module. A sequential design thus has several key components. The first is a parameter which we denote by theta. Theta quantifies the difference between E and C in terms of treatment efficacy. We next have a statistic which expresses the advantage of E over C as is apparent from the sample of data available cumulatively up to an interim analysis. We shall also need a second statistic, and the purpose of this statistic is to express the amount of information about theta which is contained in the sample. The next key component is a stopping rule. The stopping rule decides whether we have sufficient evidence to conclude the trial, that is, whether the current interim analysis should be the last, and if so, whether we should conclude that E is better than C or worse than C, or that there is no significant treatment difference. As we have said before, we shall concentrate on frequentist methods of analysis and methods which will return a p-value and point and interval estimates of theta. We first concentrate on parametrization of treatment efficacy. We shall assume here that we have been able to formulate the problem so that we have a parameter theta, and the parameter theta expresses the advantage of E, the experimental treatment, over C, the control or reference treatment, in terms of treatment efficacy. The need to specify theta is to provide a means to formulate the power requirement. So we shall assume that we need to satisfy a power requirement 1 minus beta for some pre-specified beta. That is, with probability 1 minus beta, E will be shown to be significantly more efficacious than C, assuming that there is a real difference in the treatment advantage. Significance will mean that the p-value against the two-sided alternative is less than alpha for some pre-specified alpha. So note that it is sometimes erroneously claimed that when theta is equal to 0, significant advantage can be obtained with probability alpha by 2. Our null hypothesis is specified by h naught theta equal to 0, representing no treatment difference, and we shall obtain point and interval estimates of theta. Theta is thus central to the derivation of the sequential plan, and Whitehead's recommendation is that the primary criterion for the choice of theta is clinical interpretation. We know that alternative choices of theta are possible. So in the case where we have a continuous outcome, theta could be a mean difference. In the case where we have binary outcomes, theta could be the log odds ratio. In the case when we are looking at time to event, theta could be the log hazard ratio. In each case, theta equal to 0 denotes that there is no difference between the experimental and the control treatment. However, it is necessary to check the assumption. So, for example, if we choose theta to be the log hazards ratio in a Cox proportional hazards model, it is assumed that the underlying assumption of proportionality has been checked, and that if there is any treatment advantage, then it will be quantified by the log hazard ratio. Having chosen theta, most approaches to sequential designs use test statistics which, when plotted over time, will resemble Brownian motion. We shall discuss this more on subsequent slides and see how the need arises and why the requirement is generally fulfilled by most statistics. The reason is that in many cases it appears that the log likelihood is a quadratic function in theta. Now, if theta is the only unknown parameter, then the log likelihood is parameterized only by theta, and the Brownian motion approximation can be seen to hold in many standard situations. So, consider the case when the log likelihood depends only on a single parameter theta. We can then expand the log likelihood in terms of theta using Taylor series expansion and ignore terms including the cubic and higher order terms. To this degree of accuracy, the log likelihood will resemble the log likelihood which is obtained from a sequence of the log likelihood will resemble the log likelihood obtained from a sequence of independent normally distributed random variables with mean theta and variance 1. We are assuming here that theta is small, and in this case cubic terms and higher order terms can be ignored. When nuisance parameters are present, they can generally be replaced by the conditional maximum likelihood estimates, and we again get a profile log likelihood which is a function of theta and can again be approximated using a Taylor series expansion and retaining up to quadratic terms in theta. Now, consider the case where we have binary patient response, that is, response is either of the form success or failure. If we denote by PE and PC the probability of success on the experimental treatment E and the control treatment C, then this slide gives three alternative choices of the parameter theta. So, we could have theta equal to PE minus PC, theta equal to log of PE into 1 minus PC by PC into 1 minus PE, that is the log odds ratio, or theta equal to eta PE minus eta PC, where the function eta P is given by eta P equal to integral 0 to P dt over T 1 minus T whole to the power 2/3. Now, note that the first is obviously both meaningful and easy to interpret, but it does have some limitations. So, for example, the choice theta r equal to 0.1 has no interpretation if PE is less than 0.1. The value of theta r may be set by asking for a value of PC and a desirable value of PE, but the power of a sequential procedure will be achieved for any pair PC and PE, which gives rise to the reference improvement theta r and is thus independent of the true value of PC. All such pairs should represent situations in which a probability of 1 minus beta of observing a significant advantage of E over C is desirable. Now, parametrization 1 will probably achieve this for a limited range of PC. Consideration of the third derivative of the log likelihood of theta shows that the Brownian motion approximation will also be poor except when theta is very small. So, this will mean that confidence intervals for theta will include values which are outside the permitted range -1 to 1. Now parametrization 3 is optimal in the sense that it eliminates the third derivative of the log likelihood completely, and thus ensures that the Brownian approximation will hold best for this choice. However, as you can see from the choice, there are big differences in interpretation of the significance of theta. Similar differences can also arise in the case of normally distributed random variables. So suppose that the mean response is mu e for patients who have been allocated to E and mu c for those on C. Then, assuming that large responses are good, so that we wish to have mu e larger than mu c, two possible choices are theta equal to mu e minus mu c and theta equal to mu e minus mu c divided by sigma, that is scaled and unscaled treatment differences. Obviously, there is a trade-off between the two in terms of the interpretability. The scale differences are harder to interpret. However, the distribution of the scale differences is more closely approximated by Brownian motion theory. Moving on to the test statistic, we shall assume that we have a series of interim analysis. At each interim analysis, we shall calculate a statistic Z, which denotes the advantage of E over C as has been calculated by the sample up to this interim analysis, and another statistic V, which represents the sample information about theta which is currently available at the particular interim analysis. We shall assume that Z and V are chosen in a way such that expectation of Z is equal to theta V and variance of Z is equal to V in large samples. Whitehead recommends plotting Z against V, and in that case we can look for straight line trends, and also we can interpret the gradient of the plot as the advantage theta of E over C. Some other authors have recommended construction of plots of Z over root V, in which case we would no longer look for straight line trends but would look for parabolic trends. There are two general approaches to the construction of Z and V. The first is given on this slide. So this is to use the score statistic evaluated under the null hypothesis for the choice of Z. So assuming that L represents the log likelihood ratio, Z is equal to L dash 0, in which case V is the observed form of Fisher's information, that is V is equal to minus L dash 0. Both are calculated under the null hypothesis. Now similar forms also exist when we use a profile log likelihood in the presence of nuisance parameters, and alternatively methods can also be applied to the conditional or the marginal likelihood, or in case we are monitoring survival data, to partial likelihood. Then the approximate quadratic form of L is, as we know from large sample theory, given by L theta equal to L naught plus theta Z minus half theta squared V. So this assumption, for example, leads to the likelihood based tests which have been studied by Rao. The second approach is to base each interim analysis on an estimate theta hat. So commonly we choose the maximum likelihood estimate theta hat, and the information V is taken to be the inverse of the square of the standard error of theta hat. In this case, the statistic Z can be taken to be theta hat V, so that its expectation has the required form. Now this approach is easy to implement because these statistics can be obtained directly from most analysis packages, and When it is based on maximum likelihood, the method is a sequential version of the large sample walled test. The justification of Brownian motion approximation for the sequential version of the walled test can be made by its asymptotic equivalence to the Rao score test. Our next requirement is a stopping rule. So we shall assume that we have a series of interim analysis, and at each analysis we observe values Zi and Vi based on the cumulative data up to the ith interim analysis. Now, by a stopping rule, we mean a sequence of lower limits Li and upper limits Ui. Li and Ui are functions of V1, V2, Vi, and they are used in the following way. If Z is less than equal to Li, then the trial stops, and the likely conclusion is that E is harmful, or maybe that E is no better than C. If Zi is greater than equal to Ui, again the trial stops, and the likely conclusion is that E is better than C. If Zi lies between Li and Ui, then the trial continues until the next interim analysis. So note that the conclusions which have been mentioned here have been mentioned as likely conclusions. This is because the primary purpose of the stopping rule is to determine when sufficient data have been calculated, and once the trial is stopped, we need to perform post-trial analysis to determine the direction of treatment differences. An interesting feature of this design is that it is considered to be a closed clinical trial. The design of interest for most clinical trials are closed, whereby closed we mean that stopping is certain provided that the information continues to accrue over the trial. Closure can be obtained in one of two ways. Firstly, the stopping limits can be constructed so that they converge. That is, there is a value Vmax of V such that for the first interim analysis in which Vi is equal to or exceeds Vmax, Li is equal to Ui, and the trial is forced to stop. Second, we can introduce a simple extra stopping rule in the form of stop if Vi is greater than equal to Vmax. We make a difference between convergent and non-convergent boundaries. So here we have represented some stopping rules. On the x-axis we have plotted V, on the y-axis Z, and the limits Li and Ui denoting the stopping rules. We can see here that the first two rules correspond to convergent boundaries, and the next two set of rules correspond to non-convergent boundaries. Another feature of interest in a sequential clinical trial is a double test. A double test can be formed by combining two sequential procedures. So, usual situation is symmetric, that is, a procedure with stopping limits Li Ui such that mod of Li is less than equal to mod of Ui being combined with its mirror image which has stopping limits minus Ui and minus Li. Now, the first of these will result in a claim that E is better than C if an upper limit is crossed, and that E is no better than C otherwise. The second concludes that E is worse than C if Z is less than equal to minus Ui for some I, and that E is no worse otherwise. The double test will run until both the rules have stopped. If the first finds that E is better than C, then so does the double test, and similarly if the second finds that E is worse than C, then again so does the double test. Otherwise, no significant treatment difference is obtained. Now, once we have chosen our stopping limits, to evaluate the properties of the resulting sequential design, we need to use recursive numerical integration. So, for example, if p theta denotes the probability that the sample path of the statistic z will exceed one of the upper stopping limits at or before the ith interim analysis, then this probability is given by p theta equal to summation k equal to 1 to I, probability v equal to vk and z greater than equal to uk, which can be written in terms of the probability function of z as summation k equal to 1 to I integral uk to infinity fk zk of k into d zk, where f of zk is given by integral lk minus 1 to uk minus 1 over root ik being the information, into phi of zk minus 1 minus theta ik by root over ik into fk minus 1 zk minus 1 k minus 1 dz k minus 1. So, as you can see from this integral, the numerical integration needs to be performed recursively. Now, most sequential plans use one of two approaches. In the first, we envisage an ideal continuous version of the sequential plan. That is, we assume that it is possible to obtain z continuously over time and that we have conceptually a continuous plot of z against v, assuming that v increases smoothly. Then continuous stopping boundaries, which are L as a function of v, Lv, and u as a function of v, uv, are constructed. Now, boundaries are chosen which have simple mathematical properties and usually satisfy some kind of optimality criterion. The second is based on the concept of an alpha spending function. The alpha spending function relates the null probability of obtaining a significant positive result, that E is better than C, when or before the amount of information available reaches the value capital V equal to small v. Now, we usually assume symmetry, that is L of V is equal to minus u of v, and the ratio v over v max is usually referred to as the information time. For the continuous implementation, in practice, when we have several discrete looks at the data, it is necessary to make adjustments. These adjustments can be made using the Christmas tree correction. So, the problem here is that the stopping rule boundaries can be crossed in between two discrete time points, and thus it is necessary to bring them together. One way of doing this is to use the Christmas tree correction, and it is based on a result due to sigma. It has been depicted on this slide. We shall quickly look at two related concepts. The first is of repeated confidence intervals. So, here we shall typically get a series of intervals of the form theta 1i, theta. Typically, we shall get a series of intervals of the form theta li, theta ui, such that they contain theta with probability 1 minus gamma. This will hold for each interim analysis I equal to 1, 2, etc. So, note that these are repeated confidence intervals, and these can be obtained from any non-convergent sequential design in which the null probabilities of crossing each boundary are half gamma. So, for example, the limits theta li equal to zi minus ui by vi and theta ui equal to zi minus li by vi could be used. Post-trial analysis, a stopping rule will only typically determine whether or not we have sufficient evidence to conclude the trial, and once the trial is stopped, we need to conduct post-trial analysis. So, most analysis methods are based on some kind of ordering of the possible data sets which could be observed at the end of the trial, and we wish to find the probability of observing data which is more extreme than that which has been observed. The p-value function in this case is defined as p theta equal to probability of observing data more extreme than that from the trial theta. P-values against the 1 and 2-sided alternatives are then p0 and 2 minimum p0 1 minus p0 and 100 1 minus gamma % confidence interval can be calculated as specified on this slide. Similarly, we can obtain a point estimator of theta by solving p of theta m equal to half and using the median. In this first module on sequential clinical trials, we have tried to define a uniform methodology for such trials. This is largely based on the corresponding paper by Whitehead, which has been cited in the module. A sequential clinical trial has several key components. We have defined the statistic. The statistic, of course, must be chosen based on clinical relevance and in such a way that, sequential clinical design has several key components, the first being the parameter theta. The parameter theta must be chosen based on clinical relevance and in such a way that theta equal to 0 represents that there is no difference between the two treatments, and in general, theta measures the advantage which the experimental treatment E has over the control treatment C. We shall next define a corresponding statistic, and one reason for choice of statistic is that we wish to have an underlying Brownian motion assumption hold. We have defined a pair of statistics Zi and Vi, where Zi could, for example, be the score statistic, Vi could be the extent of information. So typically Zi and Vi are chosen such that Zi is equal to theta Vi, and two alternative choices could be the score statistic and the wall statistic. We have defined a stopping rule and seen that this can be implemented in two ways. First as a continuous formulation and second as something called an alpha spending function. In practice, when we go from the continuous to discrete, it is possible to make some assumptions, and these adjustments can be made by the Christmas tree adjustment. Finally, we note that a stopping rule will only specify when we should end the trial, and in this case, once the trial has been concluded, we need to perform a post-trial analysis.